today is about preparing us for a tiny piece of work. A tiny piece of work from your grade 11, 12 piece of biology that we need to cover. But this covering and writing a test is not about, I want to take a short cut and I want to get it over and done with. It is about testing yourself. It is about slowly starting to realize the end is closer than what I think. And it is about getting myself ready. So, yes, I am going to show you a few short, important information pieces to help you for this test. But actually what I want to do is I want to invest another bit of information so that you can be prepared for the end of the year. You are going to write a test on the pages that I indicated. It will be on what is DNA, where do I find DNA. It will be how DNA can synthesize itself, and then also how proteins are being synthesized. I see a chromosome, and a chromosome is a length of DNA with a lot of genetic information on it. That length of DNA is actually the material that is called up. And if I can get to the smallest piece of DNA, I see DNA is a double helix, and I can see that this double helix consists out of a backbone with a pentose sugar and a phosphate group, which makes the backbone. But in between, I have 4 different types of nitrogenous bases: adenine, thymine, cytosine, and guanine. But they link with a complementary pair. So G always to C. And what I also see is that G and A are the larger ones. T for that, A and G are 2 rings. And we call them the Purines. And then we have the pyrimidines. Thymine and cytosine are the pyrimidine. And we see that they bond with hydrogen bonds. And there are 3 hydrogen bonds between guanine and cytosine, and 2 hydrogen bonds between thymine and adenine. Okay, and if we need to get to the other type of nucleic acid, we have RNA. We also know that RNA is a bit different from DNA. The first difference is single strand, double strand. Because this is a single strand, no complementary strings, therefore no hydrogen bonds. Then I also see that there is a uracil and there is a thymine. Right. That is important to remember. I see that the sugars differ. Ribose sugar, deoxyribose sugar. The difference is this large DNA cannot leave the nucleus. This one is small enough to leave the nucleus. When we have the replication of DNA. That is for the cell to divide and make two identical. Grade 12, it's fun time, it's play time, so please play along. Revision means I am now going to see what do I know and apply that. And then if you realize, but something is not correct, see if you can correct it, or please contact me and ask me why is how you think perhaps not correct. Okay, so let's start with inheritance, and we are going to revise all the possible inheritance theories that we did study. We are going to look at a few of the terms that are important. Dominance and recessive alleles in a gene, codominance alleles, multiple alleles, the sex-linked problems, and then what is the gender of a baby. That is a short revision so that you just can see whether you do follow the work or not. The terms that I want you to look at is haploid and diploid. A nucleus is haploid or diploid. That means that in a nucleus there can be a single set of chromosomes, meaning in the nucleus there is only one of the pair that is in the parent, one single set unpaired chromosomes, and that forms the gametes. But in a diploid nucleus there is two sets. That means both from the father and from the father and from the mother. From the father and from the mother. There is from both of the gametes fused together, and now we call that a homologous pair. Now you see a homologous pair. So, if there is a nucleus that have only one of each of the homologous pairs, we will immediately know that cell is a gamete. We will recognize a gamete that the chromosomes are single, there's no homologous pairs, there's only one set of the chromosomes, haploid. And we refer to a haploid nucleus as N number of chromosomes. That N will mean in human cells 23. You do follow that. a specific fly has 6 chromosomes. Yes, that small amount. And that means N means 3 for that fly. Diploid means 46 in human terms. Good, let's continue. Then we have our chromosome homologous pair. With this we mean that these two are similar. And I want to use that word similar. These are, sorry, let me just get. These two are similar because they have the genes for the same characteristics. It is not the same genes. We can see the gene that is on that locus is the gene for a specific characteristic, but it is not the same. No, they will be the same size and that they will be on the same location on the DNA. They will have the gene for the same characteristics. They will be the same size and they will have the same location on the DNA. In that way, they will be similar, but they are not necessarily identical. If it is an identical twin, they will be the same. But no, sorry, not even then they will be the same. They will be the same in the two individuals, but not the same there. These two are not identical. Why can they not be identical? Because a gene has a specific locus, or genes have loci on the chromosome, and the two alleles can either be the same, which means the same allele has been inherited, or it can be different. If it is the same, we say this individual is homologous for this characteristic, but it will be heterozygous for the characteristic that will play out in A. This is homozygous for the dominant feature, and here we see homozygous for the recessive feature. Good. I hope that you do follow this. Now, homozygous, dominant. Why do we have different colors? Because one is inherited from the father and one from the mother. Then, homozygous dominant, yellow, is the dominant allele. This gene, let's say it is for eye color. Both parents had the dominant allele in their gametes, or the recessive allele. Or if you can inherit the dominant allele from one parent and the recessive allele from the other parent, that is a heterozygous genotype or phenotype. Good. So let's take a problem. 2 black mice, one heterozygous, The other one, homozygous, for coat color. 2 black mice. Recessive allele is gray. What possible colors for the offspring? 2 black. So, I will start with the parental phenotype. It is black that crosses with black. The one is heterozygous, and the other one is homozygous. So, the heterozygous will be capital B, small b. And I normally will write down black is dominant, ne? And what will grey then be? The grey coat will then be small b. Important. We use the small letter of the dominant allele. And the other one is homozygous. Now there will be gametes. Immediately I draw circles and capital B, small b. So, if this is the male, this will become sperm. And the sperm can either have a B, capital B on it, or a small b, because to form gametes, meiosis take place. The homologous pair separates, yes, so there can only be one. Now I have a haploid cell. And there we have that possibility. Now show fertilization, please. And I am going to use different colors to show fertilization. So, fertilization can happen between, there's a possibility that it can happen between those two, or between those two, or this one and this one, or this one and this one. This is all possibilities. Now, each time when there is intercourse, this is the possibility that can take place. So, we can have the genotypes will be B, capital B, small b, or that one, capital B, small b. So, the genotype will be, there's a homozygous black, and there will be one. And then there are heterozygous black and 3. All of the offspring will be black. But phenotypically, all will be black. But when we look at the genotypes, there can be a homozygous black and a or heterozygous black. And now, grade 12, it doesn't matter what is the problem. Whatever they ask, you write it down like this. If they ask a genetic diagram, you have to use all of this in your answer to have full marks. They can also ask the question many different ways. Sometimes you have to do a bit of calculations, nah, but you are higher level. So therefore, you must think. Nah. So therefore, we have another problem. 2 black mice, 8 babies, yes, 8 babies, and 1 is grey. What is the genotype of the parents? Show it with a test cross. This is where I do not know the parents. I can do a test cross. So, if I have a baby and the baby is gray. Again, we have black as dominant, né? And the baby is gray, so the little baby will look like this. I write in here, small b, small b. This is telling me that from this parent, this baby inherits a small b, and from this parent, the baby inherits a small b. You agree? That is how I will start. If there is one gray baby, there is the possibility that, not the possibility, there must be a B, a gray B, and a gray B from both parents. If I look at 8 babies and 1 is gray, it means 7 is black. So black, 2 gray, more black ones, and the parents are black. So the parents must be heterozygous. Black, that gives me 1 out of 4, né, will be gray, and the rest will be black. But if I look at that, that is not the perfect ratio. But it is telling me that the smallest amount is gray. So therefore, the parents was heterozygous black, phenotypically. Their genotypes was capital B, small b, both. That is the only way how the offspring can turn out like that. Okay, now a CFTR gene codes for a specific transport protein found in cell membranes. Cystic fibrosis is an inherited illness where a mutation in the CFTR gene causes chloride ions to build up inside cells. When chloride builds up, it will change the osmotic potential. It will have a lower osmotic potential, and the water will move into that cell, which causes them to absorb more water by osmosis. Very important. In the lungs and pancreas, this occurrence leads to the production of thick, sticky mucus that can affect breathing and the production of digestive enzymes. From the pedigree diagram, what can you say about how cystic fibrosis is inherited? Explain your answer. So I have female and male, and the colored ones are the affected ones. If I look at the parents and the F1, the first generation, I again see one has cystic fibrosis, ne? The other 2 does not have cystic fibrosis. So this offspring did inherit a cystic fibrosis, and here they tell me I must use CF. So this individual, number 2, did inherit the genes. So I will say cystic fibrosis is a recessive allele. My answer will be 1 out of 3 offspring contains the disease, and even that the parents are both not affected, they do have an offspring. So 1 out of 3 is my explanation of the offspring, and the parents, although they are unaffected, did produce an offspring that has cystic fibrosis. Therefore, it is a recessive allele. Do you realize that this, I just want to show you, this is the offspring. Now, here we have another marriage. Here we have another couple. Can you see that? Important to see. That is important to understand. Here we have a marriage and the offspring. There we have the marriage and that shows the offspring. Okay. That is important to understand in a pedigree diagram. Write out possible genotypes for individuals 1 and 2. So, because there is a small cf, both must contain a small cf. Here they use the cf. We normally use only one, but I think there it's capital small, because that one did inherit a small. Now, what is the probability that individual in line 2, number 3, is heterozygous? Is there a possibility? I think the possibility is 100%. Because, no, sorry, it is not 100%. I don't agree with myself. What is the probability? The probability is heterozygous. When we test heterozygous with heterozygous, we know that the possibility is 1. I'm going back to this one. Heterozygous always gives me 1 dominant there's 1 dominant, 2 heterozygous, and 1 recessive homozygous. Now, 1 dominant homozygous. So, if I go back to that one, we have number 3 there. The parents are heterozygous, so the possibility to be heterozygous is 50% to be heterozygous. Just look, what do